So our next speaker is Manuel Mattheisen from Aarhus and Würzburg universities. And he is the co-lead for the OCD and Tourette working group and going to present an update on these disorders. Manuel, go ahead.
Thank you very much, Barbara. And thanks very much for the invitation to actually speak here. So as Barbara said, this is an update on behalf of the PGC TS and OCD working group, so the Tourette's and OCD working group. And to give you a bit of an idea who we are talking about, these are two pictures from the meetings in Orlando and Anaheim last year, the last years. And this is part of our membership. We're totaling currently some 150 individuals that are on our email lists for the work groups. And what you can also see is that we have, in terms of PGC positions, Dongmei and Nora as the lead analysts for Tourette's and OCD respectively, although Dongmei also does a lot of work and has in the past actually for OCD. Jeremiah, who is co-sharing the group together with me, and then Perry and Christy as outreach officers for TS and OCD respectively. This is where we are at with regards to the published work, and you've already seen that in Pat's slide earlier today, that for OCD, which is the right-hand side of the slide, we are basically currently at 0 genome-wide significant hits. That's with a sample size of roughly 2,700 cases and 7,000 controls and a SNP heritability on the liability scale of about 28%. And then for TS, which is on the left-hand side, and you will see this color coding actually throughout the presentation, the red for TS and blue for OCD. You see the recent meta-analysis with about 4,800 cases, some 10,000 controls, and a SNP heritability of 21%. There was a genome-wide significant hit that was reported last year. The study, however, also included a replication in an Icelandic study from Decode. And unfortunately, this locus didn't replicate in the Icelandic study, which doesn't rule out that this is a true association. However, it probably needs larger sample size, similar to the case with OCD, to robustly identify this loci. Going to the next slide, you also have seen this slide from Barbara before, so the work of the Brainstorm Consortium, Werner Jantel al. Both OCD and Tourette's were involved in these two studies. You see that they share significant genetic correlation between each other, but that especially also OCD shares a lot of genetic correlation with other traits, most prominently with anorexia nervosa, but also with major depression, for example, and other traits. And so this is why pretty early on, actually, the work group has done quite extensively studies on comorbidities, shared risk loci, and also on endophenotypes that can be identified through comorbidities. So on the upper left hand, you see a study that actually looks at the genetic correlation between Tourette's and OCD and finds that there's a difference between those individuals that have both disorders at the same time and those that only have one of the two disorders. On the upper right hand, you see for OCD and anorexia, for example, that we have done a study which, at least so far with a limited sample size that we're looking at, didn't identify any robust association with both disorders. On the lower left is a analysis focused on the childhood onset disorders, including ADHD and autism, together with OCD and Tourette's. And then on the right, as I said, actually look at endophenotypes and Tourette's that are basically, amongst others, defined through comorbidities. So for example, this class 1 with Tourette's, OCD, and ADHD, or the class 3 actually dropping OCD and only resembling TS and ADHD. We have also done a similar study in OCD that's not yet published. What this shows you is that these are polygenic risk scores for individuals that have a set of different comorbidities, or actually are only OCD. So the left-hand bars are the ones that only have OCD, then the ones right to it have a comorbidity with MDD on top of OCD. than autism, ADHD. And if it's more than one comorbidity, including OCD, it's actually the green color coded. And as you can see, when you train on an OCD polygenic risk score, you see that it's some difference actually in how that pans out or works out for OCD genetic liability, with the pure OCD cases having the highest together with the comorbid ones and the one with autism. On the other hand, this is still based on small data sets, and so we would need to see how this works out in the future. What you can also start to see is that based on these comorbidities, genetic correlations or the shared genetic liability with other disorders actually changes based on the disorder that you're currently looking at. We have heard about dimensions and the fact that we are thinking about also spectrum disorders. So this, for the Tourette case, actually in the original 2019 paper from the meta-analysis, they tried to answer a question that was already brought up in this Nature Reviews disease primers, which is that tic and Tourette's is actually the spectrum of disorders, and that potentially polygenic risk scores are distributed in a way that that there is a difference between transient tic over to chronic tic, Tourette syndrome, and simple tics, and severe and persistent. So meaning the more severe you get, the more you are at the more severe end of polygenic risk scores. And this is in fact also what they found in this study. So this is again looking into this Icelandic replication study. And what you can see here in the three different colors is A, that they looked at differences between using clean controls in terms of super healthy controls compared to population controls. But they also looked at the two different disorders or traits like tics and Tourette's. And what you can see is that there is really a change towards the more severe / more loaded end of the polygenic risk scores when they are trained on their GWAS. And the same thing, if you do that with traditional bar plots for polygenic risk scores, you see that the overall GWAS does predict TS better than tics, which is because they also have been more prominent in their sample, but that there is also a difference between the two of them. You heard about dimensions for ADHD, so we're doing the same thing for OCD. We have two working groups within our PGC work group. One is focused on OCS more broadly, and then the other one is focusing on hoarding symptoms and the hoarding dimension. It's a heterogeneous group of samples that we're looking at. It's population-based samples. It's disorder-specific samples, meaning that they have been collected for a given disorder, and that is both for OCD, but we also have a study that was originally collected for depression. And then the analyses are currently underway, and we are totaling something like 60,000 individuals for that. In terms of the instruments, there's a wide variety coming with this heterogeneous sample collection. OKR, so the obsessive compulsive inventory, is one of them, and you find the question including the subscale factors actually just here on the slide. The Toronto obsessive compulsive scale is derived from the OKR, so it's pretty close to it, but it is normally distributed, while the OKR is more distributed towards the null. There's the Padua, and there are many others actually as well. Now, as I said, the GWAS so far have been underpowered, but we're actually currently trying to solve that problem. And for OCD, we have a current freeze of for 2020, which is as of actually last week. It includes also a diverse group of samples, both from so clinical samples, population and cohort level studies, but also the biobank studies that we have heard for, and then 23andMe. Overall sample size in the end will be some 44,000 cases. That's including actually the 23andMe cases. And then the core sample, which is everything but 23andMe, that's some 14,000 cases and more than 500,000 controls, with the controls predominantly obviously coming from these biobank and population cohort studies. Additional analyses are underway, so results of that I can't show you. But what I can show you is the Manhattan plot actually of this core sample. And while we are not there yet, where, for example, ADHD was what you have seen, We are getting there. So there's one genome-wide significant hit in here, and there's a genetic correlation in contrast or compared to the meta-analysis from 2018, which just comprised of these two consortia that mostly have clinical samples. That's at 84%, and it's highly significant. Already now we have actually expressed interest for many secondary analysis once these summary sets become available, and they will become available within a short period of time. First of all, there is a lot Enigma related studies that people wanted to do. Enigma being brain imaging consortium that we have also, I think, already heard of. But there's also studies into the overlap between canine models for OCD or obsessive compulsive behavior and the categorical OCD phenotype. This is just an example of many smaller clinical studies that have collected a wide variety of different endophenotypes and phenotypes. And they will now take the chance to actually use the predictors that we can produce from the current freeze and look into their samples. And then finally, there's also evidence from some register-based studies, for example, on maternal effects and so on, that we would like to take forward and combine that with the genetic data. In terms of TS, we are a little bit behind OCD. We have secured funding, an independent R1 grant. These are the sites that are involved in this R1 and some of the other contributors. As you can see on the right-hand side, there are 4 different aims specifically. One is around GWAS and CNV analysis, and then the genotype-phenotype correlation, both in terms of comorbidities, but also sex-specific effects. A functional genomic analysis aim, and then lastly, but definitely not the least important, the imaging genomics analysis that we would like to do. We currently anticipate that there are some 14,000 cases that by the end of this year we will have analyzed with an addition of some 50,000 controls, so slightly more. And with that, I would like to close and actually thank all of our patients and the contributing sites, all the mostly uni investigators and clinicians that spent numerous hours actually to make our work a success, and obviously all the funders in individual countries and you for your attention. Thanks.
Thank you very much, Manuel. The problem that we cannot clap here, that's a pity. Are there questions for Manuel? I don't see any yet in the Q&A, but I have one to start with, which is, so when we're looking at ADHD, we have these 2 domains that show quite some genetic relationship overlap. I think for OCD, there's much more heterogeneity in the phenotype, isn't there? So is it, would you expect that if you try to disentangle the phenotype a little bit more, that you find more in terms of genetics?
It's actually a good question. So there's certainly an entire continuum around OCD, and it's not actually by chance that now hoarding, for example, is no longer part of the OCD phenotype, but it's actually a phenotype on its own. Mm-hmm. We hope that this is the case. It's part of the reason why we started these dimensional studies to actually find out more about this. But it's not clear, at least to me, whether it's similar to you, where you have actually more cases in your dimensional phenotype, but actually find less, kind of in contrast to the categorical phenotype, or whether it's the other way around. The one thing that I can say, though, is that many of the heritability studies on OCD have been actually done on OC symptoms rather than OCD. So when we go back to that literature, I think we can potentially identify that some of these factors or subfactors are actually more heritable than others. So it might be that for some of them we find more, for some of them we find less. Mm-hmm.
Okay, thanks. We have questions coming in. Do we have still time, Jerome, Chris?
Yeah, maybe for 1 or 2.
Okay. So we have 2. Historically, obsessive compulsive personality disorder has been conceptualized as being related to OCD. Have you checked if there is genetic correlation with obsessive compulsive personality disorder?
We haven't done yet. However, since we have seen that there are many biobank studies that actually have a full variety of ICD-10 codes that they can actually go back to, we hope that this is something that we can look at actually at a broader scale with upcoming analysis.
Okay. And then there's another one on how is Tourette syndrome genetically related to neurological disorders?
Mm-hmm. It's a good question. In principle, I think you can answer that using, well, Brainstorm with the caveat that the Tourette's data is rather small that's in there. But I think on average, although you might expect, since there's all brain disorders, that they're highly correlated on the genetic level, I think one of the findings of the Brainstorm paper was actually that they are not that much when it comes to neurology versus psychiatry. And I think the same is true with Tourette's and the neurological disorders.
Okay.